We now begin supplemental lecture on the cell organelles and a discussion of the cell as a protein factory. Again, we are focusing on cells as the living units of organisms. The slide shown here illustrates the DNA within the chromatin inside the nucleus, the nuclear envelope here, and the cytosol here with the events related to protein synthesis. So we start first with a discussion of protein synthesis as an overview. From the DNA, we see a strand exposed by uncoiling of the double helix. And of course, that's the antisense strand of the DNA that is transcribed to produce the initial RNA transcript that will be processed, as we will discuss, to form, in this case, messenger RNA. Messenger RNA enters the cytosol through the nuclear pores, where it encounters ribosomes, and events will occur, binding of the ribosomes with the DNA, with the messenger RNA, and ultimately the synthesis of protein through assembly of monomers of single amino acid residue that is contributed to the growing polymer of amino acids, the protein. So this overview gets us started. There are events inside the nucleus and events outside the nucleus within the cytosol, and different types of proteins that are produced and different locations where different types of proteins are synthesized. We start first then with a discussion of gene transcription. We saw, obviously, there's a double-stranded DNA, the double-stranded helix, and it is uncoiled to expose the antisense strand that will be used as the template. Proteins will bind to it and contribute to the binding of RNA polymerase, and the RNA polymerase will create polymerization of ribonucleotides into an RNA strand. That is the initial transcript. The initial transcript will undergo processing, RNA processing. Here are three examples: splicing, 5' capping, and the addition of a 3' poly A tail that we will look at through these illustrations. So in this illustration, we are seeing the structure and transcription of a eukaryotic gene. We will use as an example the beta globin gene. It's about 16,000 base pairs long. A gene contains exons and introns, intervening segments between the DNA sequences that guide ultimately what will be the amino acid sequence. So the exons are the DNA coding sequences. Contains 441 base pairs, the triplet codons for 146 amino acids, plus a triplet stop codon. There's non-encoding sequences of DNA, the intervening sequences, or introns, and they are initially transcribed between the codons 30 and 31. It's 130 base pair long, and 104 and 105, or 850 base pairs long. But they are spliced out of the final transcript. So obviously we are examining here the production of a primary transcript or initial transcript and the splicing process. It's more complex than this because there has to be cleavage, obviously, and then ligation, but collectively we are getting a splicing of the original length into a smaller length. So let's progress here then from the upper left corner. We see DNA that is coiled. Transcription involves the uncoiling, exposure of the antisense strand of DNA. Of course, the other strand is called the sense strand. The antisense strand is used to make the initial transcript. There will be RNA processing. The first example, the splicing we will see here. and ultimately messenger RNA will be produced, and the messenger RNA is the only form of this type of RNA. So nothing before this is recognized by the nuclear pore proteins and allowed to exit the nucleus. We'll talk about that in a little bit more detail in a moment when we talk about the poly A tail. And then ultimately translation will occur in the cytosol. So here we see an enlargement of the gene that we talked about. We have two strands of the DNA, the sense strand, let's say here on the top, and the antisense strand. They are, of course, complementary, in other words, and anti-parallel. So we have 5' to 3' here, and 3' to 5' here. We have a start codon here for the transcription and translation ultimately, and a terminator of the transcription, and this portion is not transcribed. So we're going to start or initiate transcription over here, and then we are going to terminate over here. So RNA polymerase will bind here. Once we get these two strands separated on the antisense strand, it'll bind and read in this direction, and then terminate and release itself when it gets there. So we can see also upstream of the, this upper one would be the antisense. Upstream, 5' upstream of the antisense start codon will be a portion called the promoter region, very important for controlling gene transcription. We'll see more and more examples of this as we go throughout these series of lectures. So the RNA polymerase binds with other proteins contributing because they have bound to the promoter region, or doesn't bind because of other proteins that are here to keep this gene transcription either slow or turned off. So those are ideas to keep in mind. So there's protein binding with initiation, and we will transcribe the exon 1, then intron 1, then exon 2, then intron 2, and then exon 3, and then the stop codon here will be transcribed. That's the stop codon for translation. So the initial transcript, or the primary RNA transcript, will contain all homologous sequence to the, or complementary sequence to the antisense strand. So we will have 5' end here and 3' end here, complementary to the strand from which it was read. So 5', 5', 3', 3', and it does complement it, but it is ribonucleotides, not deoxyribonucleotides. The initial transcript will be processed, and the first is, as we see, there are splice sites here at the ends of the exons, and that's where we'll get the cleavage and then ligation, so that ultimately exons will be ligated to exons. So we have exon 1, exon 2, and exon 3. So this is the spliced mRNA at this point, and we are going to get modifications to the 5' and the 3' ends of this spliced messenger RNA. Those two RNA processing features are illustrated in this slide. So there is a 5' cap that is added. It's a modified GTP that is added here. And then this is the coding region of the primary transcript. And then at this closer to the 3' end of the RNA strand, Their sequence is recognized by RNA cleavage enzymes. And then the RNA is cut here, and then a poly A tail is added to that 3' end. So we have these three RNA processes: the splicing we saw on the previous slide, 5' cap, and the 3' poly A tail. 3' poly A tail contributes to the stability of the message once it is in the cytoplasm, and of course it's thought to be involved in giving it permission, if you will, now to leave the nucleus. So a quick summary of things, as well as a discussion of the potential points for regulation of gene expression. So it can be anywhere from the regulation of transcriptional control, that promoter region we talked about, to the processing control for the RNA processing, transport control out of the nucleus, the mRNA stability control. So you can have inactive mRNA that might be protected from degradation. And then you have actively translating messenger RNA. So the translational control of protein synthesis occurring there, either inactive or allowing translation. And then there is post-translational control over protein function, which includes going from active to inactive states, or inactive to active states. So many levels of control of gene expression, this term gene expression being inclusive of starting with the DNA and having the functional protein. So now let's consider differential expression of the beta-globin gene family. So this would be developmental physiology if we put it in that context. We use it as an example of differential expression of a gene family, but you can get differential gene expression of an individual gene within a cell as well, in that it would be regulated. It might be very low expression at one point and high expression at another, which of course is what we're seeing if we quickly go to the graph and just see this line indicating expression. So first we'll read the legend, and then we will go through the figures in this illustration. So during human development, different members of the beta-globin gene family are expressed at different times and in different tissues. So during the development, these different members are expressed in different times and in different tissues. So this reminds us that we get cell-specific gene expression and tissue-specific gene expression. So every cell in our body contains every gene that we have, but every cell in our body does not express every gene that we have. So that's differential gene expression within a cell, or within, between cells, populations, and then you can get differential gene expression over time, developmental time, as seen here. So this differential gene expression has great physiological significance. For example, gamma globin, not gamma globulin, gamma globin. They're two completely different proteins. A subunit found, so the gamma globin is found in the hemoglobin of the fetus, which is, it's a tetramer, I was trying to think of the prefix for 4, tetramer. So there's 2 alpha chains and 2 beta, here we are talking about 2 gamma chains. So alpha 2, gamma 2. So it binds oxygen more tightly than the adult hemoglobin, as you heard me say almost automatically, the alpha 2, beta 2. And both gamma globin and beta globin are members of the beta-globin family, and this specialized form of hemoglobin ensures that the that in the placenta, where the maternal and the fetal circulation come near each other, that the oxygen will be transferred from the mother's to the developing child's blood. So just before birth, the synthesis of the fetal hemoglobin in the liver stops. So you terminate gene expression in the liver and you shift to a new tissue, the bone marrow. Cells take over, making the adult form. So it's not within one cell that we get a shift in gene expression from one family of the gene to another. It's from one tissue to another. So let's take a look at the graph here to get oriented first. The x-axis is the number of weeks after conception on the left, and then birth here with the transition from the blue to the yellow, and then the number of weeks after birth shown to the right in the yellow portion of this graph. The y-axis is percent total globin synthesis, ranging from 0 to 50. So here we see roughly before 6 weeks of time from conception, the different globin genes are expressed at different times during the human development. So just trying to orient to which. So here's alpha globin. So it rises quickly within the first 6 weeks and stays very high. Here in the solid black line is the gamma that rises quickly, persists to week 24, begins to diminish, and at birth, of course, it is very close to the level of the beta globin, and the beta globin is what will be present from birth, basically throughout life, of course. So just before birth, there's a switch from the gamma globin to the beta globin gene expression. There are other globin genes, members of the globin gene family, that are expressed very early on and diminish quickly. So it's the alpha with the delta, gamma, or the alpha with the beta that are important. We also get tissue-specific gene expression shifts. So the vertical dimensions of these shape represent the relative expression of genes in different tissues. So initially, the yolk sac is contributing to the formation of the hemoglobin, the fetal liver, then the fetal spleen, and then finally the bone marrow, of course, throughout adulthood. So a perfect example of differential gene expression that lets us point out the concept of cell-specific gene expression, tissue-specific gene expression, and regulated gene expression. Even within the cells that are expressing these genes, we get an increase, a stabilization, and then a gradual decrease, and an ultimate termination, so it's no longer expressed. So we see all these examples of regulated gene expression within this concept of differential gene expression. So what are the types of RNA? Of course, there's messenger RNA. It's used for the translation into the protein, as we just stated. Transfer RNA, we saw it, but we'll see it again in more detail. This is bringing the amino acids to the ribosome messenger RNA complex for the assembly of the multimeric or the polymeric proteins, amino acid sequences, and ribosomal RNA. So ribosomes are made up of proteins and RNA, the ribosomal RNA. So it's this ribosomal RNA that's letting us recognize a sequence in the messenger RNA and bind it and bring in the transfer RNA. And we said it's produced in the nucleolus selectively. These are produced in different areas along the chromosomes of the chromatin. So these ribosomes have two subunits and the ribosomal RNA, two protein subunits. So they're involved with protein synthesis, the translation, and they're found in two places, either at the rough ER or free ribosomes in the cytoplasm. We'll emphasize these points in more detail as we go along. So this slide allows us to make an overview of the events that are occurring. We now understand getting from antisense strand of DNA to the mature messenger RNA that now has permission to exit the nucleus. It will bind to a portion of the ribosome. We'll get assembly of the attachment of the ribosomal subunits, or assembly, final assembly, and then actual translation will occur. We see messenger RNA bound to the ribosome, the two subunits bound to each other. And then here we see the transfer RNA with an amino acid bound to it, attaching to that. Translation will occur, as we see illustrated at higher magnification below here. So we see the ribosomal components being able to bind to the messenger RNA and shift it along. As it does, it reads triplets of these nucleotides, and those will specifically bind certain transfer RNAs and thereby bring in specific amino acids for each amino acid residue in this growing peptide chain. So the messenger RNA has a three nucleotide residue codon, and then the transfer RNA, of course, has the anticodon that is complementary to that and will bind to it. So this is how we get the decoding of the DNA sequence, the gene. Then finally, there will be termination. We saw that termination codon being built into the gene itself, and it ends up in the messenger RNA. When the ribosome hits this, boom, it lets go. So we get attachment, initiation, translation, and then termination, and this frees the completed peptide. So let's talk about the endoplasmic reticulum a little bit before we talk about where proteins are synthesized. The endoplasmic reticulum is a membrane-bound structure. How many locations in a cell can you find protein production synthesis? As we said, there's really just the ribosomes that are involved with it, so we can actually reword this and say, where do we find ribosomes? Free in the cytoplasm, as we said, or associated with endoplasmic reticulum. I say associated with because they're not always bound to the ER, the rough ER. They are studded with ribosomes, and we'll look at the details of that association in just a moment. So with the rough endoplasmic reticulum, we find protein production for secretion and degradation and for membrane proteins. So three types here. Do we produce enzymes? Well, if it's an enzyme to be on the membrane surface, then yes. But other types of enzymes, as we'll see, might be synthesized somewhere else. So this illustration shows us on the panel on the left, an actual transmission electron micrograph of a cell. We see these parallel laminated stacks of rough endoplasmic reticulum, and then we see other areas of the cytosol where there would be ribosomes. On the right, we have a pictorial illustration of this. So there'd be free ribosomes in the cytosol, and then the fixed ribosomes, they're only fixed in that they stay close to and produce that studded appearance of the ribosomes. So this is rough ER with the fixed ribosomes and the free ribosomes floating in the cytosol. And the small and the large subunits of the ribosome shown here that are involved in attaching to and initiating translation with the messenger RNA. We see early translation of a message strand here and progressively later translation with termination here. So there's initiation that occurred somewhere back here as this moved along. These have already gotten on there and they're continuing the process. They're showing this as a single process, but you can have multiple ribosomes on a message increasing the rate of translation, one of those control steps for gene expression. So let's start here with number 1. We see that we've already assembled some of the amino acids. These first few are in colored yellow. That's the signal sequence. Genes that have a signal sequence in the very first exon there will produce messenger RNA that, when associated with ribosomes, will produce a signal sequence or a signal peptide. That signal peptide binds to or interacts with proteins on the endoplasmic reticulum, the signal recognition particle, or SRP, and then there's an interaction with the receptor site on the endoplasmic reticulum, more proteins, transmembrane proteins, so there's a pore here, and you see the signal peptide is attached, and then any further amino acids that are produced end up crossing the endoplasmic reticulum membrane and are pushed into the lumen of the endoplasmic reticulum. So only genes that have in their first exon a sequence for a signal peptide will target their messenger RNA for the fixed ribosomes and synthesize their proteins into the endoplasmic reticulum. So the peptide continues to grow. We already see these little blue molecules being placed on here, so we're getting post-translational processing already, such that we start with a small polypeptide. It's a growing polypeptide with carbohydrates on it already, and we end up with a released glycoprotein containing sugar groups that end up in this structure that we'll discuss in just a moment. So this process continues until the ribosome reaches the termination site. At that point, the completely translated protein is released, in this case into the endoplasmic reticulum. The signal sequence is actually removed at this point here. So somewhere along the growth of the polypeptide, that signal sequence will be cleaved by a protein inside the ER. So that protein inside the ER that does this cleavage is produced in the same way. So start thinking about that. When you find a protein somewhere in a cell, ask yourself, where would that be produced? What's the best place to produce it? Once the protein is produced inside the endoplasmic reticulum, it will be associated with a portion of the endoplasmic reticulum membrane and some of the fluid within it, such that it pinches off, and once completely pinched off, becomes a transport vesicle, or budding off is the term used here. So again, our levels of control, the translational control of protein synthesis, we talked about just a bit. We didn't really go into detail. But now we can quickly take a look at post-translational control of protein functioning. So what are some of the examples of post-translational processing? There can be folding, cleavage, multimeric formation, glycosylation, we just saw that, lipid esterification, so putting a lipid on it, making a lipoprotein, phosphorylation, making a phosphoprotein, or dephosphorylation, removing that phosphate. So, and throughout a discussion of physiology, especially endocrinology, these examples will be elaborated upon repeatedly. So what are these transport vesicles? They pinch off from the RER and it heads to the Golgi apparatus. So they really are transport vesicles. So this slide is showing us two examples of molecules, proteins that are being produced at the endoplasmic reticulum. They end up in the endoplasmic reticulum within its cisterna or lumen, and pinches off into a transport vesicle that fuses with this side of the Golgi apparatus. There is movement through the Golgi apparatus with some processing, packaging, and a variety of things like that. Some post-translational processing also occurs in the Golgi apparatus. And then it pinches off into a vesicle. In this case, we're looking at a lysosome or a storage vesicle. So this might be a degradative enzyme originating from its gene way back here that ends up inside a lysosome. Another example would be a protein that would be secreted, in this case a polypeptide, back to the gene here, messenger RNA, into the transport vesicle to the Golgi, processing, packaging, and release in a vesicle that then, under certain conditions, and we'll look at these in a moment, you'll get the movement for exocytosis into the extracellular space. So that endoplasmic reticulum. So makes us ask a question. Where is the membrane protein produced, and where are glycolytic enzymes produced? So if you're going to produce a membrane protein, a protein that had to stick in the membrane, why not produce it right at the endoplasmic reticulum where you're putting it into the membrane in the first place? Then it would just stay there. So that's where the membrane proteins synthesis is going to occur. Glycolytic enzymes, we'll see, are housekeeping enzymes, so the free ribosomes would be involved there. So whenever we encounter a protein, ask ourselves, what's the most efficient location for the cell to produce this? A membrane protein, you might as well produce it at the ER where you're pushing it through the membrane in the first place. Just leave it within the membrane when you're done, and use the fluid mosaic model to slide it down to the transport vesicle and leave it in the membrane from start to finish. Whereas an enzyme that has to act on a substrate solely within the cytosol, produce it on a free ribosome. But let's, before getting off to that, consider the smooth ER. What occurs there? There's no ribosomes, so we don't get protein synthesis. So some proteins are delivered here, such as the enzymes that are responsible for lipid synthesis. So they will undergo post-translational processing, such as activation here, to be involved in lipid synthesis. So in steroid-secreting endocrine cells, that's very important to understand. So lipid is synthesized here. Now, membrane lipids are produced in different locations. Typically, it's just outside of the membrane of smooth ER or rough ER. So we have to maintain our membranes because they are not stable forever. We have to recycle, remove the old oxidized or damaged phospholipids and put in new ones. So the free ribosomes, as we had mentioned, let's venture into this idea of rough ER and smooth ER before progressing on through this discussion. So here we see the rough ER with continuity to the outer membrane of the nuclear envelope. As we said before, that's important for producing membrane proteins that will be put into the membrane at the ER. And then if they are targeted for the nuclear envelope membranes, they simply, slither through or slip through the slippery lipids in the membrane, and that fluid mosaic model, and then end up where they're supposed to be, either on the outer membrane of the nuclear envelope, or they get to a nuclear pore and they just slip in and get on the inside. So these are being produced constantly. And then, of course, smooth ER, and we see that the smooth ER has a lot of continuities with the rough ER, so something produced in the rough ER can, if it's targeted properly, end up inside the lumen of the smooth ER and, for example, be very much involved in producing lipids. So let's think of some of those housekeeping proteins that would be produced at the free ribosomes. In a previous lecture, we talked about cytoskeletal proteins that are entirely within the cytoplasm. Automatically, we're going to make the guess, and it's a smart one, that those proteins are produced by translation on free ribosomes. Now, there are many enzymes that produce metabolic events, and they are synthesized in the cytoplasm at the free ribosomes, and they're typically referred to as the general housekeeping proteins. So, for example, those in the glycolytic pathway, we'll see this in a little more detail later, but we recall that Krebs cycle and the oxidative phosphorylation pathways have many proteins involved in them, and they are on or in the mitochondrion. So it reminds us that mitochondria produce some of their own proteins, and they receive proteins from the genes of the within the nucleus as well. So that's kind of a complex interaction of protein synthesis there, but we'll just leave it at that for now. So we've now, in our protein factory, got it produced. We got the protein produced. Let's pretend we're going to ship it out. We've got it produced in the ER. We put it in the transport vesicle, sent it off to shipping. They've put it in the box and added packing and all that stuff, post-translational processing. Maybe assembled it with other subunits, added lipids, whatever it would be doing, maybe a little bit of enzymatic cleavage, and then they'll put it in the final package after refinement, and they will then, as if you had little workers in this protein factory, the Golgi apparatus is responsible for putting it into a vesicle ultimately for its final delivery. So the Golgi apparatus is usually near the nucleus, called perinuclear in its location. Going to package and refine the proteins that come to it, and it has a cisterna. So think of a flattened membranous sac, or like pita bread. So if you open up pita bread and put your hand in there, then your hand would be inside the cisterna of the Golgi apparatus. So here we see an illustration of that. So here's the nucleus. The Golgi is typically perinuclear in its location. There is a cis face, the receiving side of the Golgi apparatus that receives the transport vesicles. We can follow the molecules inside the cistern of the Golgi apparatus. So there are these stacked pita breads that do have interconnections, so there's movement along through them. And then the opposing side, the trans face, is the shipping side of the Golgi apparatus. And this is where the vesicles are going to be produced, the transport vesicles coming from the Golgi apparatus. So we're seeing different colored spheres entering in here, little red ones and little white ones. So the red is being assembled here pretty much all by itself. A little bit of red and white there, and then only white here. So this is illustrating that refinement and processing. So the vesicles are released from that trans or releasing face, and we can see that there are three general types of vesicles: secretory granules, for hormones and enzymes that are going to be released. So these are big ones, big vesicles, and secretory vesicles, a little bit smaller, like the neurotransmitter synthesis and transport and release. Lysosomes and peroxisomes are the other example, those degradative structures that contain the degradative enzymes, or ones that produce hydrogen peroxide and very reactive species. So that's destructive stuff. If it's your factory, you want to package it into something and just don't let them float around, destroying everything in the cytosol. So let's think of a clinical correlation here. The lipoprotein transport of cholesterol across the membrane to tissue puts that cholesterol in the interstitial fluid space. Phagocytic cells will take up that lipoprotein, but can't break it down. So the lysosomes try to interact with it, just can't break down the lipoprotein heavily laden with cholesterol. The fat accumulates in those cells. They become called foamy cells, loaded with these big fat lysosomes. And then those will fuse and just degenerate, leaving the fat deposits, producing a fat streak. You get lesions, calcification, and that, of course, is atherosclerosis. So the lipid deposits can be everywhere throughout the vasculature. So let's follow this again in this illustration with focus on 3 pathways for vesicles, because we only looked at 2 in the previous example. Here we get to 3. Pathway 1 is secretory. Pathway 2 is delivery of membrane proteins. Pathway 3 is the degradative pathway. So we see different colored spheres coming into the Golgi apparatus here, a blue and a red. And as well, they're not really showing it, but it should be these little T-shaped structures also would be in the little T-shaped structures would be in these transport vesicles, and along the length of the Golgi membrane, ultimately sequestered onto a vesicle, or even included into one that's going to be exocytosed. These two sometimes are combined. The cell's going to be efficient, or it may just be doing this predominantly, depends upon the example. A good example of this would be the water pores or aquaporin protein molecules for reabsorption of water in the kidney. So there's exceptions to everything and generalizations to everything as well. But let's just follow them here. Pathway 1, get some red molecules pumped into this vesicle that are going to be exocytosed. So we get secretion by exocytosis through the secretory vesicles. There can be big ones, granules, or small ones of vesicles. Here we see a membrane, in this case, a membrane-bound vesicle loaded with membrane proteins, considering it as being dedicated to delivering those membrane proteins to the plasma membrane, or the cytoplasmic membrane. And then here, the third pathway, lysosomal enzymes, the hydrolytic enzymes, being delivered to a phagosome. So let's think of this exocytosis function of cells. There can be 2 types. Constitutive secretion, just ongoing all the time. Doesn't mean it's not regulated. Just it's constitutive and maybe do a little more, do a little less, but it's just ongoing. And then the other is regulated secretion. So remember, for every protein somewhere back here, there's a gene, so we've got to impact that gene to synthesize these proteins upon release. So the constitutive expression of a gene will be related to the constitutive secretion of a protein. Collagen is a primary example of this. The regulated secretion is the other that typically is associated with a ligand-dependent signaling molecule, receptor membrane protein, through transmembrane signal transduction. Quite often it's a metabotropic type of a pathway. We bring about exocytosis. So this is an endocrine function is a primary example of this. This is regulated secretion. So in this, when we release, we must synthesize to replace. So there's typically a parallel pathway that goes all the way to the gene, and that we'll talk about later, to increase the expression of the gene to replace what we just released. So let's keep that cycle in mind all the time. Here we got constitutive release, so constitutive gene expression. Here we've got regulated release. We're going to have regulated gene expression. Replace what you release. So these intracellular signaling pathways branch out and do a wide variety of things, especially with the metabotropic receptors. So 2 types of exocytosis, constitutive and regulated. And then we take that, extend that to the nucleus, constitutive and regulated gene expression. So this study of the membrane in the cell brings us to think of this entire endomembrane system. It's very important to conceptualize this as a single continuous membrane, because we've got membrane continuity from the nuclear envelope all the way through the ER, through the transport vesicles to the Golgi, through the vesicles, exocytosis and endocytosis with the cytoplasmic membrane. So we talked about the cycling of vesicles, exocytosis and endocytosis in a previous lecture. And we see that there is tremendous opportunity for recycling of membrane to preserve membrane as much as possible, and to endocytose a chunk of bad membrane and fuse it with a lysosome and try to degrade it. Then somewhere else you got to put new membrane, and I'm thinking of lipid component as well as the protein component into that membrane. So the lipogenesis, or the production of the phospholipids, will occur somewhere near smooth and rough ER, and that the new phospholipids will be inserted into the membrane there, so you generate new membrane, and then you lose it as you go through the system, but some of it can be recovered and recycled. So now let's transition to thinking of the cell in general as a metabolic machine. So there are two types of metabolism: anabolism, building up like an anabolic steroid. So for example, from smaller to larger, amino acid monomers to a protein polymer, glucose to glycogen, monomer to polymer. So from simple to complex. Catabolism is the opposite, breaking down, complex to simple. So you store glycogen, and then you break it down to glucose, and then you break down glucose to get ATP out of it ultimately. Carbon dioxide and water is the waste. And then ATP can be catabolized, so ADP to PI, inorganic phosphate. And of course, that's the primary chemical catabolic pathway for producing energy useful to the cell's functions. So this illustration shows an example of a catabolic type of an event that contributes to an anabolic event. So we see fuel molecules here, carbohydrates, fats, and proteins, can undergo catabolism to their metabolites, or catabolites, if you want to think of it that way: carbon dioxide, water, and ammonium. Ammonium, of course, coming from the proteins as they would from nucleic acids. And then it is not 100% efficient to produce chemical energy. In this case, it's 40% efficient, so you get heat, which is nice in cold days when you want a little bit of thermogenesis. This will bring about an anabolism to produce ATP, so ADP plus PI gives you ATP. And then ultimately that ATP undergoes catabolism to provide that energy for the energy requiring cell functions like force and movement, active transport across membranes, or molecular synthesis. So let's look at this ATP production to segue into a discussion of the next organelle, the mitochondrion. So here's the glycolysis, that glycolytic pathway we talked about, where we start with glucose, and we know there's many enzymes and intermediates along the way to produce 2 pyruvic acids. The pyruvic acids will go through another process to get acetyl-CoA, goes through the Krebs cycle, and along the way, of course, we're producing ATP, so catabolism associated with anabolism, to store, to extract energy and to store it. We produce our catabolites, or metabolites is the term that we'll just use from here on out. After the Krebs cycle, then a little more ATP, then we provide something to the oxidative phosphorylation pathway, and this is, of course, where we consume the oxygen in aerobism to produce the largest amount of ATP. So anaerobism is less efficient at producing ATP than is the aerobic mechanism. So from 1 molecule of glucose with 6 oxygens and 38 ADPs and 38 inorganic phosphates, we get 6 carbon dioxides, 6 waters, and 38 ATP molecules. So where does this occur? This is cytoplasmic. So these enzymes all along here are housekeeping proteins, and they're produced, translated at the free ribosomes. Excuse me. And these events are going to be within the mitochondrion. So the oxidative phosphorylation, of course, includes the electron transport chain that is found on the inner membrane. So again, we're segueing into an introduction of the mitochondrion. It's got two membranes. The inner membrane is shown here. It's a lipid bilayer. And we see there are integral proteins, and we get movements of electrons. It's electron transport. As it's transported, we oxidate or reduce molecules. No need to jump into details here, but it emphasizes the significance of integral and peripheral proteins being associated with each other. And of course, we are transferring energy such that we build up a hydrogen ion concentration. And then finally, the ATP synthase is driven by the hydrogen ion passing through its channel down its concentration gradient. So we use some energy to create a potential energy of the electrochemical gradient of the hydrogen ions across this membrane, and then as that potential energy is turned into kinetic energy, we can phosphorylate ADP and have some stored energy. So this mitochondrion is a pretty good guy. It's elongated fluid-filled sacs. They're self-replicating, so significant evolutionary implications there. They have their own DNA, so all of our mitochondrial DNA comes from our mother. So we all have maternal mitochondrial DNA. There is no such thing as dad giving mitochondrial DNA unless we get in there and change it all. So the outer and the inner membrane. So it's kind of like the nuclear envelope in that case. You've got something outside, something between the two membranes, and then something inside the innermost membrane. The inner membrane is thrown into these folds called cristae, and of course it's the powerhouse producing ATP. So here's the illustration that shows us that. So it's kind of a big balloon inside a little balloon. So the big balloon inside's got to crumple up. So the outer mitochondrial membrane is shown here. There's a space between them. The inner mitochondrial membrane is shown here in the kind of orangish coloration. And it has these infoldings or cristae or crests. When we look at the inner membrane at a higher magnification, we see all those molecules or some of the enzymes that we were just looking at in the previous illustration. So we see a protein. We ask ourselves, from where did this come? Some of it comes from nuclear DNA and some from the mitochondrial DNA. Just before moving on, we notice this labeling the matrix, so that liquid aqueous phase within the internal membrane or inside of the internal membrane of the mitochondrion. So many cellular subcompartments. So think of functional subcompartmentalization. It's a very important concept in anatomy, functional anatomy as well. In other words, physiology. Where does this occur? Where does that occur? How do they interact through different subcompartments for regulation of the occurrences? So finally, the intercellular junctions. Cells talk to each other, they stick to each other, they have many different things that they need to do together at the level of tissue. So tissue is made up of many cells put together, typically with a common goal or function. So you've got to stick them together, and you've got to provide them methods for communication and protecting of their spaces, intracellular and extracellular. So these are mechanisms to contribute to those goals. Intermediate junctions or zonula adherens, got to stick to something for a zone. And then desmosomes, got to have a spot weld, the macula or spot adherens. And then tight junctions, you've got to occlude movement between the cells, so zonula occludens, and then gap junction, the nexus that lets it talk to each other. So here we see them illustratively. The apical membrane, basal membrane of these epithelial cells, basal membrane associated with the underlying basement membrane, so many membrane proteins here associated with cytoskeleton, and also with the extracellular matrix. We'll take a look at that in a moment. And then we see at the apical membrane, the tight junctions are going to be formed up there. We see the desmosomes, or macula adherens, distributed throughout here. And then gap junctions distributed, in this case, just illustratively, closer to the basal. One important one that's closest to the apical is the tight junction. So what wasn't shown there was the intermediate. It's just below the tight junctions. Let's see if it is there. No, they're not showing it. So just below the tight junction would be the intermediate junction. It's about a 20 nanometer intercellular separation, the space between the cells. So we are subcompartmentalizing the extracellular space now, and it encircles the joined cells. So these are made up of microfilaments of actin. So here we see it. Let's try to get oriented to this. Here's the tight junction. And then this should be the intermediate junction, I believe, here. So let's transition over. The intermediate should be just below this. This is the apical membrane here. See some microvilli. So the tight junction is here. The intermediate junction would be just below it. It's not labeled on this slide. But this slide also lets us introduce the tight junction. So we are seeing the tight junction. The zonula occludens is going to form this patchwork of adhesions of the molecules. So they're transmembrane spanning proteins, the occludin proteins that break up the intracellular space, but more importantly, they occlude the intracellular space. So in other words, you don't want stuff, if this is your intestines, you don't want fecal material getting in between the cells and then inside the body. And you don't want your extracellular fluid escaping between the cells either. So this is a zonula occludens, the tight junction. The desmosome is next, so it's found frequently, especially in stratified epithelium. It's on the lateral sides of cells. It's a little spot weld, just helps to hold the cells together. There's also a hemidesmosome at the base of a cell for anchorage. To understand the difference between a full desmosome or desmosome and a hemidesmosome, we need an illustration. So here's the desmosome. We see cell 1 and cell 2. So for a desmosome, you've got two cells contributing each equal component. So there's a transmembrane spanning protein with a large extracellular component called the linker proteins. And then there is a plaque protein attached to those just on the inner surface. So these would be integral proteins. This would be a peripheral protein. So we saw this in an earlier illustration of membrane proteins. And then, of course, intermediate filaments are going to be attached to the plaque protein. So this is spot welding the cells. Intermediate filaments were the guide proteins, so it's attaching our cytoskeleton to the cell. Now this slide is showing us, other than the desmosomes, at the basal membrane, a hemidesmosome. You don't have the other cell, you just have extracellular matrix. So the anchoring proteins are used here to interface with the extracellular matrix proteins that are laid down through constitutive exocytosis. And then we see collagen fibers down here. So we want to use these hemidesmosomes to spot weld our cells onto the underlying connective tissue. And then the tight junction we saw previously, and this is just a higher magnification of it. So again, transmembrane spanning integral proteins. the external exposed component will bind to that of the neighboring or adjacent cell. Then finally, the gap junction, about 2 nanometer space. It's for intercellular communication. Prime example are cardiac cells. So here we see membrane of one cell, membrane of a second cell, so 2 adjacent cells. They each have multimeric or multiple subunit transmembrane proteins that have a channel. They meet with and bind to that of the opposite cell, and there is an actual channel for the flow of ions and intercellular communication thereby. So this final slide is just an example of a clinical correlation of some of the knowledge that we've gained through this lecture, and that would be gene therapy. So if we have a sick patient that has a bad gene, let's say, or a gene that isn't just right, maybe we can fix it by populating that patient with cells that have the appropriate gene, or sending the good gene or the functional gene into the patient. So here we see an isolated somatic cell from the patient that are homozygous for the defective gene. Then we get a copy of the normal allele that's inserted into some viral DNA. We get that assembled into the virus, and the isolated somatic cells are infected with the virus containing the recombinant DNA. Then the viral DNA will carry the normal allele that will insert into the patient's somatic chromosomes. And then the somatic cells containing the normal allele are cultured, and then those transfected cells with the functional gene or the normal allele are injected into the patient, and then the symptoms are relieved by expression of the normal allele. So that's an example of applying some of the knowledge that we just gained. That completes this lecture on cell and